In this demo, I will show the typical workflow of exome data analysis with SODAR and VarFish. For background, I am working on a computer inside the Charité research network with direct access to the SODAR and VarFish servers. Further, I will use my web browser to access SODAR and VarFish, and I already have the IGV genome browser open in the background and have logged into SODAR and VarFish. I will show trio exome data of the trio around NA12878 that I downloaded from the 1000 Genomes website. This data is also available in the public VarFish demo site. Here you can see the example metadata in our internal SODAR system. As a remark, SODAR is currently under development and we plan a publication and making it open source software later in 2020. SODAR allows us to store meta information for donors, samples and their experimental results using the isotype example of the donor NA12878 and their parents with the pedigree structure and the sex of the donors. Further, mass data files can be attached to the samples. For exome analysis, this would be the read alignment BAM files and the variants in VCF format. The files can be accessed through the HTTPS protocol to allow for accessing them in the IGV genome browser over encrypted connections. SODAR generates an IGV session file on the fly that can be opened with a single mouse click. IGV now asks me for my login, and after I enter it, it loads the pedigree data. I now switch to the VarFish web app. Here you can see the overview of the case in VarFish. The most basic information is the case name and list of individuals. VarFish will also display the number of variant calls that were uploaded. In this case, this includes the off-target variant, which explains the large number. Next, the pedigree information is displayed. Further, VarFish lists the databases and versions which were used for annotating the variant, and it also displays the alignment control information when available. Further, it also shows the variant control data about the variants, including the TSTV ratio and fraction of homozygous to heterozygous single nucleotide variants on the X chromosome. The Quality Control tab gives insight into data quality. The first section follows the approach of the PADI method. The first graph displays a scatter plot of the relatedness coefficient and the IBS0 count, that is the number of variants without shared allele. We expect parent-child relations to cluster at the top left and unrelated individuals, in this case the parents, to cluster on the lower right. This is the case for this dataset. The second plot displays the ratio of heterozygous to homozygous calls on the X chromosome outside of the pseudo-autosomal regions. This can be used for telling male and female samples apart. For this case, both the relatedness and the sex fits the metadata of the pedigree. There are other statistics available, such as the number of variants and number of variants with a given impact on the protein. I will not go into detail on this here. Rather, I will go on with the variant filtration by clicking on the Filter Variants button. This brings up the variant filtration form. There is a large number of filter settings that can be adjusted, but we will focus on the most common ones. First, we can load one of the presets. Say we are looking for potentially pathogenic de novo mutations in our index. We can adjust the settings by using the de novo preset. This will adjust the inheritance category dropdown to dominant de novo, keep the population frequency at the default of dominant strict. The variant impact is kept at the default of amino acid change and splicing variants, and the quality settings are set to relaxed. In summary, we look for variants where the genotype pattern matches dominant inheritance. This can also be seen in the genotype fields below, which are set to heterozygous for the index and wild type for the parents. Variants that are very rare in background populations and are predicted to change the resulting protein. Because we expect there to be few of these variants and we don't want to miss any, the quality constraints are relaxed. This reduces the required minimal depth of coverage and allows for bigger allelic imbalance than with the default quality settings. We start the filtration by pressing the Filter and Display button. After a short time, the results will appear. In this case, we get a result set of 66 variants, which is quite a lot to look through. We will now look at one variant to demonstrate the information that VarFish displays. The first three icons towards the left show membership in dbSNP, ClinVar, and HGMB public. The next columns display the variant position, the variant reference and alternative allele, the population frequency that can be adjusted, and the GNOME and PLI score. The next column shows the gene name with a drop-down of links to other databases and annotation whether the gene is in the ACMG incidental finding gene list, and HPO annotation of mode of inheritance. The next columns show the predicted variant effect and the genotypes in the samples. The last column contains buttons with link outs to Mutation Taster, the Global Alliance Beacon Network for this variant, and a button to open the variance locus in IGV, and again various link outs to other databases. Let us use VarFish variant prioritization features to help us with our analysis. I click on the Prioritization tab and enable variant pathogenicity prioritization and select the CAT score. Further, I will enable phenotype-based prioritization. Because this is a public dataset from the 1000 Genomes project, the index is healthy. For demonstration purposes, I will fill out the prioritization form with the two HPO features short stature and brachydactyly, two features of Nicolai de Breitzer syndrome. Another click on the filter and display buttons repeats the variant filtration, and this time the variants will be annotated with the CAT prediction of variant pathogenicity and the high five score between the two phenotype terms and the gene that is affected by the variant. Let us look at the results again. The first four variants have the maximal phenotype match score of 1 and a CAT score between 23 and 30. We can now go through these variants to see whether they would explain our phenotype. The way I constructed this demo query, the first variant's gene SMARCA2 is annotated with Nicolai de Breitzer syndrome. However, the variant is flagged as likely benign in ClinVar. Let us query the Global Alliance Beacon Network. The result shows one match. We can use the Mutation Taster button to assess the variant with Mutation Taster to see that Mutation Taster predicts the variant to be disease-causing, and we can inspect the huge amount of information available in Mutation Taster. A click on the IGV button will bring up the IGV instance and jump to the position of our variant. In this case, we would conclude that the variant is actually a true de novo. Let us conclude with looking at the manual variant annotation features of VarFish. First of all, we can assign color code ratings in different categories, such as the visual inspection of the IGV results, manual expert assessment of the molecular impact, whether the variant fits the patient's clinical assertions, and assign a summary rating. We can also leave a text comment. In this case, we might end up with flagging the variant to be potentially disease-causing. We can also use the ACMG assessment form to create and document our ACMG rating. Here, PS2, PM2, and PP3 apply. We end up with a rating of likely pathogenic. The next time this variant is displayed in the filter results for this case, our manual annotation information is displayed for this variant. This information is also available in the Case Overview's Variant Annotation tab. In summary, I have shown you some of the most important features in VarFish. For quality control, variant filtration, variant prioritization, as well as the collaboration features such as sharing variant annotation with coworkers.